Now we're going to look at the structures that protect the brain. First we have the skull, of course, which is a bony structure protecting the soft tissue that makes up the brain. And then we have the meninges. Just like the spinal cord, the brain has 3 layers of connective tissue which are collectively called meninges, and they lie between the bone and the nervous tissue. And just like in the spinal cord, the brain contains the dura mater, the arachnoid matter, and the pia mater. So the brain contains 2 layers of dura mater that we'll look at in just a second. The main job of meninges is obviously to provide the structural framework to the brain and protect the brain. So we see the skull, which is the hard bony tissue surrounding the brain. Right under the skull, we have the dura mater, which is made up of the periosteal layer, superficially located, and then the meningeal layer. The periosteal layer is continuous with the periosteum of the bone. If you remember, the periosteum is a layer of connective tissue on the outside of a layer of the long bones and all bones. Okay, so that's the periosteal layer of the bone, and this is the periosteal layer of the dura mater. Then, the meningeal layer is located right under the dura mater, and this is the proper brain covering. The dura mater is really thick. If you remember, we talked about how thick the dura mater is. It is as thick as the rubber glove you use in the kitchen. Now, the dura mater is rich in a lot of blood vessels. One of the main blood vessels that you see over here is the superior sagittal sinus. The superior sagittal sinus drains blood into from the lateral aspects of the cerebral hemisphere. Okay, so this region over here is rich in blood vessels. Under the dura mater, we have the arachnoid mater. The arachnoid mater contain these extensions. If you see, these are called the arachnoid villi. The arachnoid villi, like the microvilli, but these are called arachnoid villi, which are projections that protrude through the dura mater into the sagittal sinus. Okay. The strong projections all the way into the dura mater are called arachnoid granulation. The functions of these granulations is to absorb cerebrospinal fluid directly into the venous circulation system. So these structures that are hair-like structures, along with these arachnoid granulation, help in absorbing cerebrospinal fluid during the circulation of the cerebrospinal fluid through the brain. And then we have the pia mater. The pia mater is the innermost layer of the meninges. Now, of the 3 layers, the meningeal layer is the layer that is continuous with the spinal cord, and obviously the other layers are also present in the spinal cord, but the periosteal layer does not continue into the spinal cord. Now you see this image over here. The images shaded in green are the cranial meninges. What you're looking at right now is the dura mater. If you peel the dura mater, you can actually see the gyri and the sulci in the brain, and you can see that the meninges surround the skull all the way around, all the way down the spinal cord. So you're currently looking at the this is the superior view of the brain. Yes. Oh no, that's not the superior. That's the anterior view of the brain, and this is the lateral view of the brain. The brain contains 4 chambers known as ventricles. There are 2 large lateral ventricles. we have, so these are the lateral ventricles over here, and these lateral ventricles form an arch in the hemisphere. And then we have the interventricular foramen. The interventricular foramen connects the lateral ventricles to the third ventricle over here. So these are the lateral ventricles, which are the largest chambers, and this interventricular foramen bridges the gap between the lateral ventricles and the third ventricles located right over here. And below the third ventricle, there is another channel known as the cerebral aqueduct. The cerebral aqueduct. The cerebral aqueduct leads to the fourth ventricle of the brain. And this is important because this is where cerebrospinal fluid is actually produced. Okay, so this over here, you see the anterior view of the skull, and you see the lateral ventricle, which forms an arch, and this is the interventricular foramen, and you see the third ventricle as a very thin line in the back, and this is the cerebral aqueduct that connects the third ventricle to the fourth ventricle. Okay. Alright, so this is the figures from A&P Reveal from your book, and these are the lateral ventricles. When you're looking at the well, this is the superior view of the horizontal section of the brain. So these two chambers are the lateral ventricles. So these are the lateral ventricles. The lateral ventricles are the biggest ventricles. And right over here, you see the third ventricle. And then you have the fourth ventricle, which is all the way to the bottom. Now, this is a beautifully cut section. This is a superior view, and you can see how large the lateral ventricle is, and you can slightly see a really tiny third ventricle, but you don't see the fourth ventricle and the connecting ducts in this picture. So the ventricles, these are four internal chambers within the brain where we have structures for producing cerebrospinal fluid. We have two lateral ventricles, one in each cerebral hemisphere. And the interventricular foramen is a tiny pore that connects the lateral ventricle to the third ventricle. And another little duct known as the cerebral aqueduct connects the third ventricle to the fourth ventricle. Now the fourth ventricle connects to the central canal of the spinal cord where we find the cerebrospinal fluid. Okay, and then we have the choroid plexus in these structures. Choroid plexus is a bunch of specialized blood capillaries, and these are basically invaginations or foldings of the pia mater into the ventricles. These choroid plexus contain special cells known as modified ependymal cells. You may want to recall what ependymal cells are. Ependymal cells are neuroglial cells, okay? And these ependymal cells, along with the capillaries, form a structure known as the telacordia. The telacordia. The telacordia is the structure that is responsible for formation of cerebrospinal fluid. Okay, so these choroid plexus are made up of modified ependymal cells and capillaries. So this is a picture of the choroid plexus. The choroid plexus line the ventricles, and The choroid plexus contain specialized ependymal cells, which are a type of neuroglial cells. The ependymal cells play an important role in production of cerebrospinal fluid, and if you remember, their surface, or the apical or top surface, is covered in cilia. Cilia are brush-like structures, so they are long hair-like structures which beat back and forth, and they are helpful in circulation of the cerebrospinal fluid along the central nervous system. And in addition, the ependymal cells also contain microvilli. If you remember, wherever you see microvilli, you will find a lot of absorption. So the apical surface that contain microvilli help in absorption of cerebrospinal fluid. The cerebrospinal fluid is present in between the arachnoid matter and the pia matter, and it's present in between the subarachnoid space, is what is the space between the two is called. It is a watery clear fluid, very similar to plasma, but it contains way less proteins than the plasma. Now, the cerebrospinal fluid contains more sodium and chloride than the plasma, but it contains very less potassium, calcium, and glucose compared to the plasma. So the osmolarity of the cerebrospinal fluid is very different from the osmolarity of the plasma. And this needs to be constantly maintained, and any change in the osmolarity of the cerebrospinal fluid, or the pH of the cerebrospinal fluid, can be very damaging to the brain, because it can cause changes in the biochemical reaction in the brain, and it can also cause drastic changes in the intracranial pressure, and the brain cannot withstand any major change for more than 5-6 minutes. After that, the tissues in the brain starts dying, and the person can either die or enter into a coma. So the brain produces about 500 ml of cerebrospinal fluid in a day, and normally at any given time we have about 100 to 160 ml of cerebrospinal fluid present at one time. We have about 40% formed in the subarachnoid space in the brain, and about 30% is formed by the general ependymal lining in the brain ventricles, and about 30% by the choroid plexus. The brain has three main functions. The first one is to provide buoyancy. The brain on its own weighs 1500 grams, but in cerebrospinal fluid it weighs only 50 grams. So the brain applies a lot of pressure on the floor of the cranium, but when it floats in cerebrospinal fluid, it makes it buoyant. So it kind of floats in the cranium, and that's why it's able to rest on the spinal cord and the nervous tissue without actually damaging it. The second function of the brain is to provide, let me get the pointer, a protection from trauma. So if you've heard of shaken child syndrome or concussion, this is when the jolt is really severe. The brain tissue strikes with the inside of the cranium, which physically damages the brain. Now, this normally doesn't happen. You literally need to take a sledgehammer or a baseball bat to somebody's brain to actually severely damage it. But on a day-to-day basis, the cerebrospinal fluid can protect the brain from severe jolts, which is on a day-to-day basis. Okay, and then it provides chemical stability. The pH of the cerebrospinal fluid is about 7.32. And the main job of the cerebrospinal fluid is to clean the metabolic waste from the nervous tissue and regulate the chemical environment. And any change in composition of the cerebrospinal fluid can severely damage the nervous system. For example, when there's concentration of glycine is really high, it disrupts body temperature and pressure. Okay, and if the pH is too high, it causes dizziness and fainting. And if there's not enough oxygen to the brain, it can cause ischemia in the brain, and a prolonged deprivation of oxygen to the brain. When I say prolonged, I mean 5 minutes, which is not a lot, but just even 5 minutes of oxygen deprivation to the brain can cause irreversible damage to the nerves in the brain. The normal cerebrospinal fluid production rate is 0.2 to 0.35 ml per minute, and most cerebrospinal fluid is produced by the lateral in the lateral ventricle by the choroid plexus. And now let's look at the normal route of the flow for cerebrospinal fluid. Now the cerebrospinal fluid mostly is produced in the lateral ventricle. So in this picture, let's look at the major structures. This over here is the lateral ventricle. Everything in red that you're seeing is the choroid plexus. This region is the third ventricle. This thin duct is the cerebral aqueduct that connects the third ventricle to the fourth ventricle. The fourth ventricle has two apertures. One is the lateral aperture, and this one is the median aperture, which allows the circulation of cerebrospinal fluid. Yeah, so the cerebrospinal fluid is secreted by the choroid plexus in the lateral ventricle. The cerebrospinal fluid flows through the interventricular foramina into the third ventricle. You see the third ventricle has more choroid plexus. So more cerebrospinal fluid is added in the third ventricle, and the cerebrospinal fluid flows down the cerebral aqueduct into the fourth ventricle. The choroid plexus in the fourth ventricle adds more cerebrospinal fluid, and then the cerebrospinal fluid flows out of the lateral and medial apertures, and it circulates back into the brain. Okay, and through the median aperture, some fluid enters back into the brain for circulation, while other goes down the spinal cord so that the central nervous system gets surrounded with the cerebrospinal fluid. Okay, now during circulation, at some point the fluid reaches the arachnoid villus. Now if you remember, the arachnoid villus are knob-like projections which are present in the arachnoid matter, and they extend all the way to the dura mater. So what do you think is the function of arachnoid villus right over here? Now the arachnoid villus act as, you might want to write this down, a one-way valve for the flow of the cerebrospinal fluid into the venous blood. So this is the way cerebrospinal fluid gets out of the brain and mixes with the venous blood. And the force that helps this happen is the hydrostatic pressure, because if you remember, the osmolarity of the cerebrospinal fluid is different from the osmolarity of the blood. So if you remember, the suprasagittal sinus that's present over here has a different concentration of protein than cerebrospinal fluid. So this creates an osmotic gradient which allows the outflow of the cerebrospinal fluid into the veins. Now, this is important. Why do you think this is important? Take a minute and think about it. We are constantly producing cerebrospinal fluid, and we need fresh cerebrospinal fluid every time, because cerebrospinal fluid basically carries all the waste products from the metabolism in the neural cells in the brain, and it needs to go out somewhere somehow, right? So it's taken out via the arachnoid villus. Now, if there's an obstruction in the path of cerebrospinal fluid absorption into the arachnoid villus, the cerebrospinal fluid cannot be absorbed, and it will lead to a pathological condition known as hydrocephalus. Now, hydrocephalus is a condition wherein the brain accumulates abnormal amounts of cerebrospinal fluid. When this happens, the fluid, of course, you're constantly producing fluid. It needs to get out somewhere, but it does not get out because of obstruction. So it creates a lot of pressure in the cranium, and when the cranial pressure gets disturbed, it can cause a whole lot of developmental anomalies in babies, or in adults it can cause brain tumors, or it can be caused by brain tumors, but it can cause infection in the brain. So if you have too much accumulation of cerebrospinal fluid, the pressure is going to be eventually it'll be too much, so much that it will press against the other neurons in the brain. Now, in babies, if you don't allow the brain to grow freely, it can cause a lot of developmental defects. Now, some of the most common causes for hydrocephalus can include infections like bacterial meningitis. Meningitis means inflammation of the meninges. This can be either due to bacteria or due to certain viral infections, or it can be due to trauma. Even brain tumors can obstruct the outflow of cerebrospinal fluid, leading to hydrocephaly, or accumulation of cerebrospinal fluid in the brain. So we talked about another condition over here. We just talked about it. Meningitis. Meningitis is the inflammation of the meninges, and this meningitis, now the causes we talked about can be bacterial or viral origin, but meningitis causes severe headaches, stiff neck, fever, nausea, vomiting, seizures, and coma. And the moment a person is infected with bacteria or virus that causes meningitis, it can cause tremendous damage to brain and even death. And often this is irreversible if it's not treated well in time. And the only way we can detect meningitis is by doing a spinal tap. Okay, so a spinal tap is where they're going to draw some cerebrospinal fluid by injecting in between the L3, L4 lumbar vertebrae, and do some tests for diagnosing the presence of bacteria in the cerebrospinal fluid. And when a person is diagnosed for a bacterial or viral meningitis infection, you have no time. You have to start treating the patient immediately to prevent death. Now, at any given time, the blood in the brain receives about 15% of the blood, and it's important because the neurons have such a high demand for energy, so they constantly need glucose and oxygen consistently, because even a slight interruption can cause loss of consciousness. In about 4 minutes, if you go without receiving oxygen, it can cause irreversible brain damage in coma and death. Okay, so the brain although it requires a constant supply of energy and oxygen, it also brings a lot of antibodies and macrophages and bacterial toxins and a lot of harmful agents. So that's why we have a blood-brain barrier. So if you remember, the blood-brain barrier are mainly formed by astrocytes. So these astrocytes reach out with their perivascular feet. So if you don't remember what these are, you might want to go back to nervous tissue chapter and take a look at it. So these perivascular feet basically reach out to the endothelial cells, and they basically seal off the junctions to seal the gaps in between the blood vessels and brain. This ensures that anything that passes into the brain must pass through the astrocyte and not just sneak through in between them. Okay, the endothelial cells, on the other hand, so there are two types of cells. The other cells are endothelial cells. The endothelial cells are more selective and can exclude a lot of harmful toxins that are entering the brain tissue. So we have a lot of specialized cells, endothelial cells, at the choroid plexus. Why? Because choroid plexus is the region where cerebrospinal fluid is formed. So So at the choroid plexus, the brain is highly protected by the barrier using tight junctions. Okay, so there are a lot of tight junctions that do not allow stuff to pass through easily. These tight junctions are absent in other ependymal cells in the region because it is important to allow some fluid exchange between the brain tissue and the cerebrospinal fluid. Okay, so the cerebrospinal fluid is the blood-brain barrier is highly permeable to, so let's see, permeable to water, glucose, and then stuff like oxygen, CO2, alcohol, also caffeine. And that's why alcohol and caffeine affect your brain very easily, right? So the blood-brain barrier is very easily permeable to alcohol and caffeine and substances like nicotine in cigarettes and also anesthetics. So these are the stuff that can easily pass through the blood-brain barrier. Some ions like sodium and potassium and chloride can easily pass through. Some waste products like creatinine can slightly pass through the blood-brain barrier. So these are things that need to be taken out of the brain, right? So these stuff can easily pass through. Now, what cannot pass through the blood-brain barrier? If you think of it, antibiotics and other drugs. So these cannot easily pass through the blood-brain barrier, and that's why it makes it highly complicated to treat brain diseases. Okay, so trauma and inflammation can damage the blood-brain barrier. So when a person undergoes trauma, they're at high risk for infection in the brain. Now, the blood-brain barrier is not present everywhere in the brain. It is absent in the third and fourth ventricle. So the cerebrospinal fluid in the third and fourth ventricle is in direct contact to the central nervous system neurons, and this region has centers known as circumventricular organs. These circumventricular organs contain specialized structures which act as receptors for monitoring blood glucose, pH, osmolarity, temperature. So these structures or the receptor in the circumventricular organ are extremely sensitive, and this is important for us to monitor and maintain homeostasis. But the fact that we don't have any blood-brain barrier in the third and fourth ventricle makes this region vulnerable to virus infections like the West Nile virus or HIV virus that can cause meningitis.